So there is some cheating for the back rows. Again, this is mostly meant as an interactive part. If you want to completely participate, if you really want to get a feel of what we're doing up there, there are still some seats in the front. We have also at least doubled the number of participants that registered by now. Okay, so let me get started by saying welcome. My name is David Grunwald. I'm tech part of the imaging group, and we've been doing a lot of discussion over the last 2 years of what do we need to do to make images more comparable. What this is a presentation where we tell you and show you to the extent we can what this group had come up with, and was written into documents, went through the different gremia and the different meetings, and has been agreed on. So this is no longer something where we are saying, this is something we could do. This is something where we say, this is the perspective of what we're going to roll out over the next year. There are parts to it that are ready. You can order a part, you can go ahead. There are parts that are under development, but what I want to do is give you an idea of what it is and why it might actually be something you like. So let me start by doing something you shouldn't never, ever do. Put coffee on a microscope. Okay. Having done that, we have a very small number of slides, and we'll upload them later, so please don't go through the pain of writing anything down. There's a couple of papers that came up over the years, and there is a large file on the 4DN wiki for the imaging group that has about 100 pages of things you can read up on if you want to get a little bit more background and if you want to get a little bit more of a taste what can be done in quantification of images. Some are listed here. They're the ones that are basically the examples to what we do here. But motivating, I want to start with something that just happened yesterday, right? During this meeting, we all started understanding, okay, there are the projects that we started with, and then there is what we could do together. And what we could do together is really interesting. And there is also this idea of discussing what can we do. And one of the big parts is image the same genomic loci or feature in different cells in activated states with multiple imaging modalities. This is labeling techniques, this is microscopes. And then basically the idea here is we want to look and say which one is best, right? Now, there are many, many aspects to best. Some are about, is this label suitable and doesn't interrupt the cycle or the process we're looking at? Another one is, which one gives us the most information? Which label gives us the best image? Here's a problem. How do we know? Is there a metric? Is there something that we can say, oh yeah, we just take this image and we run it through, and then we know this image is better than that image, right? Would be nice if we had. Well, problem with images is a lot that we're in an interstate between science and arts, right? We see things. We can absolutely say that by perspective, there's one ball bigger than the other, and we all know it's not. It's the same circles, the same size, it's just the impression. Well, if we look at data, this is not going to run because it's a PDF, but if we look at this, there's filters and imaging filters that say, well, this should be very clearly a signal, but what about over here? Yeah? What are we looking at? Can we introduce quantification for it? Now, the dominant method in the thinking and the discussion around all the different problems that we have became single molecule FISH. Single molecule FISH because it is at this interlevel where, yes, what you get are diffraction limited data. It is a well-established method. It should be establishable in every lab. For record, in my lab, students need half a year before it works for them. But I have been in I don't know how many meetings with students of mine, with students of other people, with other PIs, with whatever, friends over a beer, looking at images and then this question, oh, this is a signal, right? But why is this not? Yeah? And the outcome of these discussions is normally, well, If I put the threshold a little bit to the left, I have no signal left. And if I put a little bit to the right, I have a lot of signal left. Now, why is that a problem? The problem is that a threshold from the theory part should be something where you plateau, right? You have large change, you have a region where it's stable, and this is the area where you threshold reasonably. If you have something like, so, well, it's meant to have everybody up front here, so I only talk to it for the camera. Okay, so if you look at thresholds like these, in single molecule fish, we often look, the intensity distributions look like this. Yeah, and you make a best guess. So what I'm saying is, it's a good thing to think about it as one of the methods that should work, but we still have enough problems. Yeah, I had somebody saying at our poster yesterday, saying, well, you know, I never know what's a better picture. Like, if there would be a way to tell, it would be really nice. Now, coming back to pictures over here, it's all true signals. It's just a question of signal to noise. Yeah, it's how much background is there, how does it come above. But yeah, at this level, this looks worse to me than that. It does have more photons, less background. So in that sense, what is better is something that is really a question that drives us. Which should bring me towards the end of my introduction. One thing we've had to realize over the discussion, especially when we realized, so what happened is last year we sat down and said, okay, let's get a common sample. Jan Allenberg provided us nicely with nucleoporins stained with this super bright, super good staining method that they can resolve individual spikes. And we sent it out to many, many labs and imaged it and collected the images, and then came together and looked at the images and wanted to make some sort of a statement of there's confocal data, there's wide field data, there's super resolution data, and we looked at it and was like, yeah, we really can't, right? But there's something else we can. And that is we can look at the workflow of what it is we do. Well, what we do is we basically have a lot of different components in our experiment until we get to having some data to analyze, right? Dave Gilbert and the Cell Standards group have been doing a hell of a job creating SOPs that try to address this part, right? The labeling, well, how many of you have had this experience of learning a labeling protocol from somebody and you do it right next to them at the bench and theirs works and yours doesn't? Sound familiar? Happened to me many times, happens to my students all the time. Yeah? So there is variance here. Preparation. Well, let's talk preparation for a moment. So here, typical things to prepare with, right? This is just cover glasses. Fisher brand. Pretty good glasses. 0.15, 0.17, whatever the spec is. Okay. It's more comfortable, right? Glass bottom dish, high resolution imaging, different glass. Now, just as a little side comment, if I have an objective from a certain company, let's take Olympus 20X or Olympus 60X or whatever. This thing has a specified function, right? It's a piece of engineering. It should do something to a certain degree. And we assume that the labels that are on there tell us something about it. There's a magnification, there's a field number, there's a numerical aperture. Okay. There's also an expectation of how good the optic is. But Olympus didn't use these. Wait, go ahead. And they buy their cover glasses from a company called Assistant in Germany. And that's the only glass that would actually give you the performance that that objective is made for. And it's the same for Zeiss, it's the same for Leica. So there is specificity, there is a need for having a look. This is variance. It doesn't mean you can't use this cover glass. It doesn't mean you can't use this guy. They're great, right? You don't always have to go through the trouble of drawing it on a glass, mounting it in a metal chamber, putting it on your scope. Like, sometimes if you want to push it, maybe, but it's variance. Then the 4-pi microscopes hit the market. Now, the lab where I was hosted during my PhD, they got one of those. Undergrad students spent the whole summer measuring thicknesses of cover glasses. And not of these, not of cheap ones, the good ones. Just because it's an interference method, 20 micronanometers thickness difference make a hell of a difference. Yeah? Okay, so we looked at lots of parts of variance, but that is still a machine. Yeah? In all the different parts that we have on this little graph here, this one is the most well-designed, most tightly built to specs piece that we have. Now, talked with Mr. Rajas last night. He was like, in all this, we have this on our poster, right? In this whole line of events, if I talk with colleagues and friends, the biggest variability, the biggest difference that we see are where? Right here. That's crazy. That's what this is about. We don't have to have a variability there. And I think if we do work to get that better under control, we have a much easier time looking at our data comparable. And then it also provides us a leeway into a different age of data processing. Now, here's the only little problem. There's a good number of people in the field, and they have my respect. I don't really disagree, but they say this can't be done. There's no way. This has been tried before. You shouldn't. It's like, how do you do that? Because, well, what you would need is you would need to put a power meter somewhere. You need to take some measurements, and that's going to go wrong. Like in an applied physics class, most likely 90% of the students won't put a power meter the right way into a microscope. So there's that sort of concern. But there is some movement coming. Zeiss and France is running the last year a prototype testing with a company called Argolite. We've discussed the standard. It's not that easy to use, but it's a pretty nice standard. The only problem is it has a $5,000 price tag. So you still need to know what you're doing. Leica and Zeiss in Germany are talking about a German industry norm, how to avoid it, and if you can't avoid it anymore, what to put into it. So what is it we're looking for? What is there to a microscope? Well, in the end, the hope is that my little pipette here and that machine have something in common. For this guy, I have a clear expectation of precision, right? Half a unit of a scale. That should be my error. Something like that should exist for my microscope. And where does it come from? Well, there's, for one part, the quality of the image. Now, this is something that companies love to talk about, and that's something how we try to judge pictures. Quality, impression, look. This is the arts part. Yeah? But there's definitely an impact it has on our measurements. Now, the other component that there is that if I move this little slider back here, at least for one's front here, you should see there's a lot of light, right? And maybe in some cases I need less light, but there's light going in. And then on the other side, well, there should be something like light coming out. But, where is the signal, right? Well, let's focus that a little bit. And voila, there are lots of beats. But apparently it does look much, much better with more light. Now, we're a little unfair here, right? If I just make this a little darker, my big picture looks better because I get some room light out with my hand. Now, this is variability. This is part of where is my microscope located, what is the room like? We don't write that into metadata, right? Interestingly enough, the camera that records the image sees the difference very clearly. So photons, in that sense, must be a quantity that is important to us. Which parts do we have of photons? Well, we have a source that they're coming from. We have a path that they take to the sample. We have a number of them that go into the sample. We have a number of photons that come back from our fluorescence, how they travel through the system, and then what the detector is doing with it. And these are really the two quantities. If we get our optical performance and our photon flux straight, we can say a lot about a microscope. Now, side note. In the discussion that we had over the year, there has been more than once the situation arose where somebody felt like we're threatening to say, but your microscope isn't good. That's not what this is about. Yeah? This is about knowing how good it is, or tracking it over time, knowing, is it doing the same thing today that it did yesterday? And if my colleague in Rob Singer's lab, who does this protocol very nicely, sees 300 RNAs plus minus 25, and my colleague in Raja's lab, who does this also very, very well, sees 500 plus minus 50 RNAs. Could it be that there is a difference that comes from a microscope? These are the questions that we're going after. We're not going after, oh, this is good, this is bad, this is, you'll see. Yeah. Okay. Now, this is where I would ask Max if you could come. We'll set up a little experiment about how to generate data and then go through some explanation with it. I think we need to reconnect the power to the stage. Now, this is a picture, and this looks bad, right? Like, these are little dots. They're all about 500 nanometer spheres. Yeah. And if we look out of focus here, there seems to be some sort of a spherical pattern back here. This area looks somewhat okay. So what's going on here? Well, using point sources like this is one of the best ways to question or to probe your optical performance. And so basically, what we're setting up now is a Z-stack. You ready? Where all we do is we say, okay, let's take a number of steps that are a certain distance away from each other, right? And get a start button. And you see on the oscilloscope in a moment, hopefully, that there is a signal of triggers, and that makes the stage or the objective go up, and we see how it steps through. Okay, that most people somewhere have heard about. But what can we learn with it? And how do we get these data out, right? That's part one of our deliverable. And it is basically a program that is around. It's an ImageJ plugin, where what you do is you take a step like this. Yeah. You see this curvature? I mean, I should be ashamed of this microscope, right? This is like, oh my God. I can tell you we made an effort to get it this good. But what do we see here? What would we guess? Where is the problem here, right? Whoever wants to know, please stay afterwards. I can show you. But How do we analyze this data? Yeah, let's go for a report, right? Max, could you open your program and start loading data? No, please after just. Okay, so now this is not news. This is 50 years old knowledge. The problem is that for most labs that do not come out of a trade of dealing with the question of what is my image doing, it's pretty hard to extract the data. Now, there is a program that you see here. It runs an image.j, and it's basically an easy way to analyze beat images. And what the consortium is saying is, for some data that have a certain aim, want to do a certain measurement, we would like you to add this sort of data and these reports as metadata to your experiment. So all you do is you basically load them. Yeah, you can follow Max along here. Yeah. Yes, please.
Okay. So the NOP lab made this, by the way. It's in Java. It's free. You can use it. And we were quite impressed with it. It's very simple. You take these stacks. I have loaded 2 stacks here in different colors. And once you've loaded them, you specify a thing or two about your microscope. For instance, what the wavelengths were of the fluorophores that are in these images. And then you enter some information about your microscope. And it's very basic: the magnification, the NA of your objective, the pixel size of your camera, and so on and so forth. This is really all that you need to enter. And once you've done that, the program will try to detect the beat in your images. And you can vary the threshold a bit to see that you're indeed only detecting beats above the background and not some kind of noise, because it depends a bit on what data you put in, whether it has a high signal-to-noise ratio or low. But as long as you can see your beats and you adjust these sliders, that's all there is to it. Once you hit next, it's going to spend some time calculating. And since we don't have all that much time, I'll just cut to the chase with one of these reports that we made yesterday evening in a hotel room while setting this up. And the great thing about the reports that this thing spits out is that you, get an idea of how good your microscope is. I don't know if we have time to go into how this works, but the idea is that you get a couple of figures on how big the size of your beads was apparently in the picture. And by the information that you've given about your microscope, the program knows how big these beads are supposed to be. If they're much bigger, that means your microscope is blurring them out, and it's probably not performing as well as it should. So these figures give you an idea of that on the whole, as well as an idea of whether your sample is put level or not. In this case, it was clearly not, as you saw going through the stack, that the focus changes over the field of view. But also a couple of other parameters such as asymmetry, which means that the spots on average aren't completely round, which usually means there's some kind of optical aberration. And the nice thing here is that it also gives you an idea of how these properties vary over the field of view. Some parts in your field of view may be better than others. And here you clearly see, for instance, for the planarity, that there's a clear tilt in one direction over the field of view. Now, because this tilt is so incredibly big, it masks the other properties quite a bit that might occur here. So I have a report of another microscope that was in slightly better shape, where here, for instance, I won't go through all of these, but for instance, here you see that for the maximum size of the beads that has been detected in the images, that this size is a lot smaller in the center of the field of view versus the edge of the field of view, which you would expect because most microscopes have been made such that the optical quality is best in the center. If it's not in the center, though, that indicates that your microscope has some trouble and might need servicing. What you can also do if you do this for multiple channels is look at how good your co-localization is, if these channels actually overlap to the extent that you think they do. For instance, here you see that there's apparently a fairly even amount of shift in the XY plane across the field of view. But if you look at the individual components of that, X versus Y, then we see that it's really only in Y that you have an even shift, but in X and Y, it varies quite a bit from the left to the right whether your spots actually are overlapping, if they overlap, or that there was actually a small distance in between. But these are just some examples of things you can find with these reports, and the main thing that we want to convey here is that A, it's a good idea to see whether your microscope does what it's supposed to do, and it takes relatively few time to find that out. But also that if you don't want there to be any question about whether your data was recorded on a microscope that functioned properly at the time, why not generate one of these reports and submit it with the data that you took? It only takes half an hour, and you can prove that your microscope at least performed to a baseline where you could say, well, this is actually two overlapping spots and not an artifact. So it should take a large amount of the variability away and give you a nice piece of paper to tell your service representative that maybe next time you're going to pay for your microscope service, you want them to do a slightly better job. I guess that's what I have for PSFJ. I'm sorry? If it does point spread functions as well, you say? I think it just fits two-dimensional Gaussians, so it only does take an asymmetry measure and compare it to the diffraction-limited case. You do get average beat analysis over the field of view that you could use to do further point spread function analysis. I don't think that's implemented in this version of the software.
But it leads us directly to the next part, right? How will this be implemented? So DCIC says what they would like to see is that they get this PSFJ run on their side, and they don't want the report from you, they want the data set. Now the data set that we just recorded, this data set does contain PSFs, right? All nicely in there. You can just extract them from the data set. You give it with, if it matters for your analysis, you're most likely in a tier of experiments, and we'll get to that in a moment. There, this will be asked for. Okay, now this comes with one reminder though. These slides, well, we have a cover glass, right? And we have a mounting media. And these are not your cells, these are not your samples. So whatever that report tells you is the best case. It doesn't get any better when you start doing your experiment. Yeah? It is good practice to use data like this and the analysis of your data to introduce corrections, to introduce some morphs, to align your channels in a dynamic manner so that you get more reliable in what you have. But what these data just told you in the report is, well, be careful. Ask yourself, do I see more overlap of signals out here? Because if I calculate my overlap by pixels that overlap, then, well, the signal's bigger out here than it's in there. So there's a bias here. Yeah. Now, okay, so the argument is being made that if you have Z-stacks, you should get the same signal width. And while that is a nice hope, the problem is that the signals are physically different in size in different parts of the image. So even if I project them, they stay larger. They're just larger. The objective does not produce a uniform function. What I see depends on where in the field of view I see it and what the condition of the sample is. Yeah? So some of the things that got me into a faculty position is to figure out how to get such data from the living cell so that I can do the registration on data that I acquire from my cell while I take the rest of the data. That might be overkill for many, many things, but this gives you an estimate. And it allows you to see what happened after you had a student who went around like this. This objective is no longer the same. Absolutely not. If you have a high-NA objective, something that's really important, really expensive, this might already change its function. Yeah? So what happens if a stage runs into it. These are things you can monitor. But now, we have a limited amount of time, and what I really want to say here is, this is just telling you something that is out there that you can do, that we suggest you do. In order to do it, there is a tetraspec kit. You can buy this. It's like $200, $300. They're good for about a year, depending on how much you use them. They can last a little longer, but the mounting doesn't do it for 10 years, that much I can promise you. And take bead images. Now, let's come to the interesting part. Let's come to the part where last week, Wednesday to Friday, we've been at a spectroscopy meeting right before this. So all people that build spectrometers, microscopes, and the real question that we have as a problem, right, is, well, what do we do with this component? This is a component that can become messy. Because, well, how do you measure that? This is a picture from a paper from Rob Singer's lab. Rob doesn't pay much attention here. Okay, so what this is about is, well, this is the cone of light that you get out of here. Yeah, this is what you deliver into your objective. Now, your objective will not release that easily. And there's another problem. This microscope here has a bandpass filter, right? Bandpass filter is something where you have a function like this, and every out here is blocked, and the light in here you take. But they have bandwidths, normally 40 nanometer. Yeah, there are filters that are called long passes. This can go easily be 200 nanometer. So the light changes its color. It's one of the big problems in measuring light. And what we basically tell you in this paper is, well, if you want to do that, you need to take a power meter that has a thermo head. It's also not news. It's just news to our community. And then install like a profile, get an idea of what your dream profile looks like. It's involved. Doing that can easily take you a day when you know what you do, and the first time that you figure it out, you'll be busy a little longer. Now, I would say there's a rational fear that the consortium says, well, but you have to do that. Yeah? Thankfully, they're all pretty reasonable people and say, no, that's too much, there must be a different way. So the different way is the project is the part of what is in progress. Yeah? Max, where are the tubes? Tubes are over here. Oh, okay. On the table. Ah. So I'll give it a round, at least in the front here. So these are tubes that you can basically, that we're developing. They can do certain things already. They can't do other things yet. Now we're in alpha test phase. We've been to Andy Bauman's lab. We've been here to NIH to get an idea of what it is we need to do with them to make them applicable. Now you basically mount them instead of your objective. And then what they do is they're sensors for photons in, photons out. They can measure photons and they can generate photons. They can do that over different wavelength bandwidths and over different power levels. And that is important because just as a little question of curiosity, does anybody have an idea how much light this is? Like, please come here, have a look. Anybody? Rob, come on. Rob Coleman, hey, come. Brian, come, have a look. How much light is that? From here, it looks like it's like 10 watts. 10 watts. So we have 10 watts. What do we get here? 8 watts. Okay, let's see. Mr. Rieger, we have another absolute expert here. 30? What unit? Watts. Ha! Okay. Well, we talk about something that's in the back here. This is the most cheap and useless form of a fluorescent light source. This is standard, yeah, standard like mercury.
And you want the total, what units are you talking about?
Yeah, what is the right unit? The question is. No, we want to know here at the sample, like at this objective, how much power is going into this objective and put through this objective? How much light? I mean, there must be a value for that. Let's measure it. Yeah. Jörg, please, why don't you come here? We have a power meter head and we have a power meter. Want to get started? Text sometimes is not that easy, right? So you see, what I did basically is I called some of the developers in this room out to give you an idea of why this is not trivial. Well, the answer is that's about 40 milliwatts. But it's on a very small area. The field of view of this objective, of what we illuminate, is not that big. We're still talking about power densities that are kilowatts per square centimeter in the 100 range. This is enough light to immediately arrest any sort of translocation of the things that Andy Baumann is looking at. Because, like, if you wonder why this matter, talk to Andy about what he thinks about the difference of this much light and this much light. By the way, what do you think? How much less light is this now? It's still 10 watts, okay. Anybody? It's a TEMF, it's an OD10 filter. For my eye, it looks a much bigger difference. And that's a thing. My eye is the worst possible detector I can throw into this. Yeah? Now my camera on the other side. Yeah, if we go back here, this guy doesn't care, it's linear. That thingy counts. Yeah? Clear ramp. Like, we set this threshold a little bit. Yeah? We just look at beats in here. And you see this is terribly distorted. Yeah? Terribly distorted, but there's a point function here. Okay, now this test tube, of which I misplaced one by now. Oh, yeah, here. So this little machine is basically going to help you integrate this data. And it does so by having two modes where you have one function with a diffuser. So this is how it generates light. When it generates light in a linear ramp. If anybody wants to see how linear it's on the power meter, like, please come by after the talk. Then the other part is where you have the sensors. Yeah. Okay, so these are the two modes that it will produce for you. It's going to have about 3 different sensors in there, and that makes it to some degree smart. Now, we can't promise anything and everything. What we're going to give you is a way to calibrate your microscope and get an idea of the photon flux. We don't have the calibration values yet, so I can't tell you what the variance is. We'll figure that out. But it also has the ability to measure what your imaging protocol does, right? Light on, light off. So for whatever we can with the information we get, we will try to make your life easy in the sense of, well, maybe the easiest thing for you is run your standard imaging protocol, and then the tube is going ahead and just trying to figure out what it's supposedly seeing and which data it can extract from there, so that afterwards you only have to do a few measurements. Yeah, it is have an interface to a computer. It's wireless, so it can be mounted inside something like an OMX machine. And we will roll it out with a roadmap that is approximately saying, well, we have the annual meeting, 2017. We have annual meeting 2018. We have spring, which is where the beta version should come out. We have summer, where we're going to ask more labs for collaboration and testing. And then in autumn, around this time, should be available for distribution. Cost estimates are still cost estimates. I can promise you it's not going to be $5,000. The version that we have right now is in the range of more maybe like $200 to $500. We'll see what happens once we send it out for production. But it's going to be in the range of reagents that you normally use. So your eyepieces, if your microscope is built. So we use lasers. This is our tissue culture microscope. Yeah, we don't do much science on this. But lasers are much easier to measure than lamps. And I can do that for you as well. And because it generates area, you can use it either for flat field detector like a camera, or use it for a confocal system. Doesn't make a difference. We're pretty sure that Warren, this is a variant of something that Warren Zipfel and Ted Young built in the past. So also this is not completely new. 50 years ago, somebody had the idea of putting in a calibrated light source. 20 years ago, somebody had the idea of putting in a calibrated light source. Here we are again and say, put in a calibrated light source. The question is, how hard is it to do? Yeah, but I mean, how hard can it be? But yes, it's going to work on any sort of system. That's the other big take-home message that we as a working group had this year. When we looked at the nuclear pore data from all the different microscopes, the only level at which we could say something comparable is the raw data. Is what is written in the file, what is the photon amount? Because afterwards, there's so much proprietary processing, there's so much different analysis, it really becomes a, yeah, this works well on this sample. What else do you want me to say? Yeah. Now, I want to use the last few minutes of this to pitch one more idea. Oh, sorry, I forgot something. Now, we do biology, right? Sometimes I would like to know something about whether this person looked at me at the time. So I just took a picture, right? Documents some sort of behavior. It might be relevant as a supporting data set to my paper. Now, how much do I need to know for this picture about light income, light outgo, optical aberrations? Anybody has any strong feelings? I know Jörg had, because Jörg was the person who said, no way, there's so much we do where you don't need all this bullshit. Okay, so what we did is we've basically sitting down and scratch our heads saying, well, what is it what people do? And what are the recommendations that we give you for your microscope? And that is what we call a descriptive level 1. Yeah? Large-scale effects, transfection control, bacterial plate, yeah, lab group picture, yeah. Those are things that you take, they're there, you're all good. Yeah. Okay. Then there's the next level where you go in and you say, well, but I do want to know something more, right? I want to know if I count all the DAPI-stained nuclei in my image, and then I want to see how many cells have a transcription site or a DNA FISH probe signal. Then you quantify, you do some counting, right? Quantification, counting, it's pretty much equal, right? So then we go one up and say, well, okay, if you do that, please tell us a few things, like what is your optical sampling? Yeah? What source of light did you use? But we don't ask you to measure. We change our demands for once we go into single molecule FISH, once we go into something that is small, once we go into data where your threshold is no longer rock solid plateau. Yeah? So once you start seeing in your data that, yeah, there's a high dependency on my sliders, and I don't really know where to set it. Once you start saying, well, I want to count the 3D distance between two spots, or, well, I know they're in 3D, I just count them in 2D because I Z project, because 3D data processing is really beyond what I can do here right now. But if you start doing these sorts of analysis, it's time to document what you did and how you did it and how well it was done so that we can compare it. Now, single molecule fish is a good example for data quality for a wide range of what happens in this consortium. This is what most people face. There are some groups that want to push it further. And if you want to push it further, the first and easiest thing you could do is say, oh, I do this in live cells. Believe me, it's a different animal. We haven't done much more than saying, okay, you are in trouble. Please give us more data. And please do this much more frequently so that we get an idea of what the variances are here really in the experiments on a day-to-day basis. And then there's people like me who think, oh yeah, we have a tissue culture microscope. Why don't we build a Z drive so we can show Z steps of a meeting? If we go ahead and we build something new, or Jörg's lab, if you come up with something that hasn't been done before, please be really clear and collecting everything you can. And that's basically what these levels mean. They're all published, they're on the wiki. And we've had that not that long ago, I think Paul Kaufman, are you here? Yeah. We had this discussion with Joan and you about what is this measurement? And is this a D1 or a D2 or is this an A1? Where does this go? And I acknowledge this right away. There is not an absolutely sharp line. Yeah? What this is guidelines to see where you are and then make your best guess as a good scientist of what would be the right thing to do. With that, is there an added benefit? Because everything I told you so far is, well, please reserve some of your time or have a student who puts some beats on a slide. What could you get something else out of here? And what I think is a vision of, wouldn't it be nice if my single molecule fish data would generally come with something like, they have 2 sigma reliability and a better 90% detection efficiency, or we're more than 90% certain that this is a signal, and then we take it. And that's the signals we count in an image. Yeah. Does anybody have such data on their single molecule fish? Right, we don't. But it would be nice, right? It'd be really nice if we could tell our computer to say, hey, 2 sigma, 3 sigma, this is my sort of comfort level, that's how certain I want to be. At that level I want to count, and then I want to count at a certain, okay. So, yeah, you can do that. And what sort of benefits does it give you? Well, tracking, live cell, right? Here's something that's called a multiple target tracking or a scale approach. You say yes, no. Here's an approach where you say probabilities, and you just say, oh, this is rather unlikely, but these two are pretty okay. And you can start inferring information from there in a quantitative way, in a way where you say, oh yeah, I have a movement of a particle that I see with around 75% probability the frame before and 90% the frame after, therefore I infer. Or you go to the military and take some of their technology for these sorts of decision making. It is out there. To do that, though, what you need to have is calibration of your detector. And that is a big, big, big thing that you get here. Yeah? When you record the photon detection, you record with this test tube ramps of intensities that allow us to calibrate the detector. And so we go from counts to photons. Photons have a Poisson statistics, photons have properties that we can use to do analysis. The detector has properties we can use to do analysis. Out of a sudden, we can make estimates like here, we have Bernd Rieger again here, we have your Beversdorf here. Yeah? That's where I push the limits of saying, hey, this is a machine. And actually, we know what it does. We can measure what it does. Let's use this extra information and pull and push more data out of our data. Down here, just as another example of why is this a good idea for fish. Well, if you look at these images, these are beads. And what we did is we used a white light laser and just produced noise. Yeah? And the question is, Well, which method finds how many of the particles and how many right and wrong? Well, you basically see, yeah, there is differences. There's differences that in one case is defined. In the other case, we need to squeeze the methods to basically work within a certain performance interval. What else? Well, Storm has been brought up. Super resolution images are on the desk, right? Now, there have been a couple of papers, one pretty extensively dealing with the question of how do you measure resolution in super resolution images. If you talk to the authors, some of them around here, they will tell you, no, this is not perfect, but it was a first damn defined measure. Again, it needed detector calibration though. You can't just do this on, oh, I have an image here, what's the resolution? But it's a very helpful thing because it's a number we can compare. It's a number that we can extract. You know? So there's a direct value here. And then there's one other thing. I can't thank you enough for the Z-stack. Now, RNAs like Rob's lab, Rob Singer's lab, and I did my postdoc, a lot of single molecule in fish, right? And it were low expressing RNAs. So yes, you could project the images. It was okay. You had a very low probability of accidentally putting two things on top of each other that didn't belong to each other. But you go into live cells, you look into something that is much more abundant. And all of a sudden, this is no longer such a good idea. Yeah? And then you start coding, or you find somebody who coded something. You look for the right plane and say, oh, this is the brightest one, so I take this as a one, and then try not to use the off-focus signal. Well, why don't we use the off-focus signal? Why don't we do 3D fits? Yeah? This is all possible. It's pretty possible. You can do it with all sorts of probability estimates again. And then you also can deal with your compartments, right? Because your nucleus, unfortunately, in one plane can look much smaller at the top than it looks in the center plane. Yeah. So once you start dealing with questions of how do I wrap a surface around a compartment that may be not even labeled completely, these data become very, very handy to you. Yeah. And now here's my take on it, but joint analysis will eventually have to come up with joint data processing SOPs. And we're just providing one piece of towards the goal by saying, look, we know what you can know about a microscope. Why don't we record it? It's not that hard. That's the end of my pitch. Be aware, this is rolling out. The beads, as I say, they are available. If you haven't seen the document, look it up. If you have questions about it, please write to Joan or Rob or me. We can definitely communicate that again over the consortium. Start looking at the tables. DCIC has to reach the point where it is mandatory to have this data. It's going to be a sliding process over the next year to the next annual meeting. Yeah? There's more metadata that you need, and the imaging work group hasn't been able to completely nail down what values there are. There's a good catalog. I hope we can agree tonight. David? Okay, and I want to say thank you to my lab because I can't do this on my own. And if you look at the test tube, you have to look at Max. That is really the advantage of having somebody who loves electronics. If you look at our PSF reports, Matthias has taken most of that data. And then the reason why we do that is people like Li Chin here actually doing science. Because we love our technology, but we do also do science. Thank you so much. Yes.
So, for example, because I'm running like rare, so I do run units on the software and I'm seeing so we do fish for the So the way how I look for overlap, so I'm using the same software. The question is, if I provide the images, which I've done like in Metamorph, and they were corrected for the overlay to another software, would usually you have the same result, or it depends on the software?
So variance and data analysis, depending on software packages, it's a question where you can only burn your fingers. there are some software packages that tell you better than others what they actually do to correct. If you take deconvolution and 3D adjustment, like Huygens is one of the cases. There's Metamorph, they tell you much, much less. There are a couple of others around. I basically went through, I don't know, 5 years of talking to every damn sales representative on any conference I could find at a booth, trying to figure out, can I use your software? And sometimes it took me 3 years to understand if or if not. There is a change, though. Like in this consortium, you have people that do write software. Yeah? Yeah, Jörg Beversdorfer is here. There's a couple of other people, and I think that is something that might also be a very good joint analysis thing to say, well, what sort of data analysis pipeline do we want? What is the standard we trust? And if there are new features, is there a way that we can feed it in? Yeah? We have a badly extreme amounts of software to deal with many, many, many cells. This whole GPU processing field, like I think there is something to do here. But if you remember that slide where it said, oh, here's a microscope, and this is what we can control. Well, the real struggle starts after there. Jörg, yeah.
I first I want to say thank you, David, to you and your group for putting a lot of effort in writing this up, presenting this in a digestible way. But can I ask for one more kind of addition, like to make it even more digestible? Would it be possible to get like a 1-page or so write-up of like pretty much the key points, like which beads do you recommend, where can I buy them, what's the order number, here's a short protocol, how I make a bead sample, you know, like I think something very short, something really easily accessible without actually going onto the 4DN webpage and then clicking down 5 levels or so and go through the 100 pages you mentioned.
Send it around by email. That would be my preferred version.
Yes.
I can tell you that, what's the number on this thingy? T14792 at Invitrogen, Fisher, whatever you call it nowadays, will get you a slide that has beads in different sizes. They're all multicolor beads. And you have a ready sample to go. You can buy that right away, and you don't need to prep beads. You don't need to deal with the hassle of getting used to how to deal with beads. They're stored at room temperature, and you can carry them along and put them in boxes and ship them wherever you want to go. I really, really, really suggest you have that. Now, while we're on it, I use this to point out one other little effect that is often overlooked. There are other beads. Like where bead. Yeah. No. Sorry. This is for the people in the room who feel like, oh God, I don't really trust my microscope and I have no idea what that facility guy is talking about. Focal check beads. Yeah, they are a very, very beautiful thing in cases like this. Just let me show you. Come here. Because on this one I can play this movie. So these are focal check beads. Yeah, and they give you an idea about what happens if you go away from the very surface. Because one of the biggest limits that you have with these these samples, they're small. They're within a micrometer or 2 micrometers of your cover glass. They do very well. Yeah, these things are much heavier. They don't fall on the cover glass. They stay 10, 15, 20 micrometer deep into the immersion. And all of a sudden, things start looking very different. So this is a little comparison. This is a turf objective used to do a bead compared to something that is a little bit better corrected. And basically, the shapes are substantially different. The heads that you see, yeah. But just keep this little movie in mind. That really helps you visualizing very fast without running any report, getting an idea of how careful you want to be. Yeah. But yes, I'll take your comment. We will use the genius of drone. As of today, like Lisa basically said, everybody who has a microscope in the group needs to have somebody here. As of today, you know this order number, these are the beats. Go. Yeah. And it's on where we make one side summary. I hear you. As soon as I did the summary for Rob for tonight. Andy.
It's going to be great to check our microscopes as often as we'd like to tell our students to check the microscopes. It's wonderful. Thank you.
Yeah, no, thanks so much. I mean, chromatic aberration is another thing that you see here, right? The rings are not the same size with different colors. So one of my standard track committee issues is that I sit down with a student and say, well, this is not the same cell, right? Yeah, it is. No, well, when it's not the same plane. So yeah, it's really helpful. Thank you so much. Appreciate it.